My name is Abdulazeez Isah, Chemistry Department, Faculty of Science, University Putra, Malaysia. The topic of my presentation is acute toxicity of sodium taurocholate via zebrafish mortality, behavioral response, and animal metabolomic analysis. Sodium taurocholate is hydrophobic bisalt. Because of its hydrophobicity, it is detergent in nature. Like every other hydrophobic bisalts, their accumulation and prolonged retention due to the impairment of their bile duct could result in the cytotoxicity in hepatocyte, that is liver cells, and also epithelial tissue of the intestine. This might lead to dissolution of the membranes and damage of the important organelles, causing apoptosis and necrosis. There are cases of cancer which are related to high level of hydrophobic bisalt. Example, cancer of esophagus, stomach, pancreas, small intestine, and colon. Alteration of microbiome in the small intestine also by hydrophobic bisalt could lead to various disease conditions. It is because of the cytotoxicity of the bisalts, hydrophobic bisalt, that this research is conducted on the sodium taurocholate. The aim of the research is to determine the lethal dose LC50 of the sodium taurocholate. It is also the aim of the research to determine the effect of the behavior, the effect of this salt on the behavior of the fish, and to also determine the effect of the sodium taurocholate on the metabolisms in the fish. It is the aim of also of the research to determine the biomarkers responsible for the change, and also to suggest the metabolic pathway through which this change occurs. The method adopted is immersion methods, where the different concentration of the salt, of the sodium taurocholate is prepared, in which the fish are exposed into for 96 hours. And every 24 hours, the mortality of the fish will be recorded. It's recorded. And the surviving fish after 96 hours exposure are subjected to metabolomics and behavioral studies. In the behavioral studies, we use viewpoint technology, which generated matrix data and numerical data, plus movement path of the fish. For metabolomics, standard procedure are used to prepare the sample, and then the spectra of the sample is acquired using 500 MHz NMR spectrometer. The FID file produced is processed and binned for multivariate analysis, which we use SIMCARD for that purpose. The result of the test shows that sodium taurocholate does not have any significant effect on the weight of the fish, because the weight before and after, there is no significant difference. But converting it to the percentage increase, it shows that the percentage increase in weight of the highest concentrated fish shows to be quite lower than that of the control and the lower concentration. The LC50 were calculated only at 72 and 96 hours, because at the first 24 hours and 48 hours there was no data recorded. The activities of the fish is categorized into three: large activities, small activities, and then inactivities. For the large activities, which involve higher rapid motion, the rapidity of the fish is reduced by the drugs, because in the highest concentration we can now see that the rapidity is reduced. Then for small activities, there is no significant difference between the control and then the lower concentration, but for the higher concentration, there is a significant difference because that of the higher concentration, small activities is higher. For inactivities, it shows that at the highest concentration, the fish tends to be inactive. Duration for the activities tend to correspond with the somehow corresponded to the kind of the activities. This is the movement path, tracked by the the computer as uh, the fish move during the test. The red lines represented large activities, the green line represents small activities, and then inactivities are represented by the black dot here. So as we can see that the red lines, is reduced as the concentration is increasing, showing that the rapid, the large activities of the fish reduces with increase in concentration of the salts for both 1 minute and 10 minute record. Uh, the total distance covered also goes down as the concentration of the salt, increases. Here also the speed is reduces with increase in concentration of the sodium taurocholate. This is the result of the multivariate analysis. We have the PCA here, which shows the preview of the variation which exists among the samples of the fish. Here we have the PLSDA, which shows a clear discrimination between the control sample and then the treatment group. The treatment group. The summary of it here shows the validity of the results as the values actually have confirmed that they have confirmed the goodness of the fit and then predictability of the model. The validity of the model is further confirmed using the 100 permutation test, which also shows a very good intercept values for upper and lower intercepts. The difference between the metabolism of the control and the higher concentration here is determined using the OPLSDA, and then the goodness, I mean the summary of fits, which is also generated, shows that the model is very good because it has a very good value. From the OPLSDA of the control and then the highest concentration, we were able to generate the BIP. The BIP gives the list of the metabolites that are significantly affected by the effect of the sodium taurocholate. Here is the S plot, which gives the upregulated metabolite on the right hand side and then the downregulated metabolite on the left hand side. The loading column plot here also discriminate between the upregulated and downregulated metabolite that are affected by the change. Those one pointing upward are upregulated, and then the one pointing downward are downregulated. This is the list of the metabolites, which shows the chemical shift, and then the fold change, which is the number of times the concentration of the metabolite increase or decreases. This is the logarithm values. The negative values are those of the downregulated, and the positive values are the upregulated values of metabolite. Here is the plot of the logarithms, values. Those values above the baseline, are the values of the metabolite that are upregulated. Then the ones below the baseline are those ones of the downregulated metabolite. Then we have the plot of the pathway impact. The pathway impact here start from 0 to 5. Those pathways that has a greater the pathway impact are those ones that we suspected that the change occur to them. The conclusion, it is concluded that sodium taurocholate is toxic only at higher concentration, and then it takes some time before the toxicity effect begins on the fish. Then non-lethal concentration also at 750 and 375 lower the activities of the fish in terms of the total distance and speed. And The 750 milligram per liter also of the salt has been able to trigger a change in the metabolism of the fish by upregulating taurine, creatine, acetate, and so on. It also downregulated glucose, glycerol, glycine, and many amino acids. Thank you so much.